Welcome class. In this session, we'll be discussing regarding nitrogen metabolism in detail. So where we'll be discussing about how this nitrogen will be uptaken from the atmosphere and it will be fixed in case of plants. So first we'll start with it of the nitrogen cycle. So this nitrogen cycle, it is a type of gaseous biogeochemical cycle. So which will be operating in a ecosystem or in the environment. It is a gaseous biogeochemical cycle. And this nitrogen cycle, in case of nitrogen cycle, the atmospheric nitrogen, it will be fixed through various means in order to make it available for the plants. And this process of conversion of nitrogen into available forms such as ammonia, so that is called as nitrogen fixation. And this nitrogen fixation, it can occur through two ways. One is biological fixation and another one is the non-biological nitrogen fixation. So non-biological nitrogen fixation, it occurs through industrial as well as the electrical fixation, whereas biological fixation, it involves the participation of microbes. So microbes, they favor the biological nitrogen fixation. So in case of industrial and electrical fixation, so here the industrial emission, whereas in case of electrical fixation, the energy that is generated due to the lightning as well as the thunderstorm. So this will be helpful in fixing the atmospheric nitrogen into ammonia, whereas in case of biological nitrogen fixation, certain microbes will be there which will convert this atmospheric nitrogen, or they reduce the atmospheric nitrogen into ammonia. So once this ammonia is formed, so this ammonia in the soil pool, it will be converted into the ions such as nitrites as well as the nitrates. And this nitrates, once they are formed, they are radially uptaken by the plants. And in the plants, they will be assimilated in the form of amino acids or in the form of the proteins. And once in the plants, this nitrogen is fixed. So as animals are dependent on the plants as a food source, so they will also consume this plant biomass and the nitrogen gets accumulated in the animal biomass. So once the animal biomass and plant biomass accumulates nitrogen, so how this nitrogen is cycled back to the atmosphere? So that can be explained through in the three different steps such as ammonification, nitrification as well as denitrification. So we'll look into those steps in detail one by one. So first step is ammonification. So here the process of ammonification, it can be defined as, it is conversion of the amino acids or the proteins, amino acids or proteins into ammonia. It is called as ammonification. And this process of ammonification, it will be favored by a group of organisms called as ammonifying bacteria, or these are also called as, or the ammonifying organisms or the ammonifying bacteria will be there. Similarly, the decomposers or the heterotrophic organisms also, they favor the process of ammonification. So here what happens is, so this bacteria or the ammonifying microbes, they will act on the dead remains. So they act on dead remains of plants and animals. And in case of this dead remains of plants and animals, nitrogen will be present in the form of amino acid. So these amino acids, they will be converted into free ammonia. So they will be converted into free ammonia by the activity of this ammonifying bacteria or the decomposer. So it not only includes bacteria, certain fungi, they also play important role in the process of ammonification. So they will act on the dead remains of plants and animals, and they convert this amino acids or the proteins. They cause the decay of them, resulting in release of ammonia to the soil. So once this ammonia is released into the soil. So it will be converted to the ionic forms, and this process, it is called as nitrification. So that is, next step is nitrification. So this nitrification, it can be defined as, it is a process of conversion of ammonia into nitrates or the nitrites. Nitrates or nitrites. So that is called as nitrification. And the process of nitrification will be catalyzed by the bacteria called as nitrifying bacteria. And this nitrifying bacteria, it includes Nitrosomonas, similarly Nitrococcus, and then we also have Nitrobacter, so which are involved in the process of nitrification. So these enzymes, these bacteria, they will be possessing enzymes called as nitrate reductase within them. And this nitrate reductase, so it helps in conversion of the ammonia into first the ion called as nitrite. And this nitrites will be later converted to the nitrates. And this nitrates will be present in the soil pool or in the soil solution in the ionic forms. And this can be uptaken by the other plants, and it can be assimilated by the other plants as the amino acids or proteins. Whereas there are certain other soil bacteria also where they have the capability of conversion of this ammonia or nitrates directly into the atmospheric nitrogen, and that process is called as denitrification. So that is called as denitrification. So in case of denitrification, so it is a process of conversion of ammonia or the nitrates into the atmospheric nitrogen. So that is called as denitrification. And this denitrification will be favored by the denitrifying bacteria. So these denitrifying bacteria are soil anaerobes, or these are the anaerobic soil organisms. These are anaerobes which are present in the soil, such as Pseudomonas, similarly Clostridium. So these are the examples of certain denitrifying bacteria. So these Pseudomonas, Clostridium and all, they act on the ammonia or the nitrates that are present in the soil, and they release the atmospheric nitrogen, or they release the nitrogen gas to atmosphere, and this process is called as denitrification. And this denitrification can also occur through the industrial emission or the thunderstorm, lightning and all, but it is majorly favored through certain anaerobic soil microorganisms such as Pseudomonas, Clostridium, which are commonly called as denitrifying bacteria. So these are the three steps that are involved in the cycling of nitrogen back to the atmosphere. So that is what is explained here in the nitrogen cycle. So the decaying biomass of plants and animals, they undergo the process of ammonification, nitrification, and some of them, they also undergo denitrification so that atmospheric nitrogen will be released back. So that is regarding the nitrogen cycle. And in case of nitrogen metabolism, so in the next part, so we will be discussing regarding biological nitrogen fixation. So we know what is nitrogen fixation. It is conversion of atmospheric nitrogen into ammonia. So that is called as nitrogen fixation. And suppose if this process is catalyzed, or if this process is favored by the living organisms or the microbes, then it is called as biological nitrogen fixation. So it is defined as reduction of nitrogen to ammonia by living organisms. So it is called as biological nitrogen fixation. And in case of biological nitrogen fixation, The microbes involved are called as nitrogen fixing microbes. And this nitrogen fixing microbes, they may be free living in the soil or they may be in symbiotic association with that of the other plants. So example for free living nitrogen fixing bacteria includes Azotobacter, Bigerinkia. These two are the free living aerobic microbes or the bacteria, whereas free living anaerobic microbe include Rhodospirillum. So these are the three free living bacteria, and majorly they belong to a group called as eubacteria. Those are involved in conversion of nitrogen into ammonia. So that is regarding the free living organisms. So next is symbiotic organisms. So this symbiotic organisms includes rhizobium as well as frankia. So rhizobium fixes nitrogen in case of leguminous plants. So they help in nitrogen fixation in leguminous plants, whereas frankia, it is helpful in nitrogen fixation in case of non-leguminous plants. So leguminous plants mainly includes the pulses such as pea, beans, similarly groundnut. So all those comes under leguminous plants which belongs to family Fabaceae. They form the root nodules, whereas non-leguminous plants include ulna, similarly casuarina. So all these are the non-leguminous plants. So in non-leguminous plants, root nodules will be formed by the frankia, which will fix the atmospheric nitrogen, whereas rhizobium, they fix the atmospheric nitrogen in case of leguminous plants by forming the root nodules. And here, how exactly the root nodule formation takes place by the activity of rhizobium. So we'll see in this part. So here, in case of rhizobium, so it will be having certain genes or the factors within them called as nod factors, which codes for the enzyme nitrogenase. Similarly, in case of plants, they are going to the roots of the plant, they are going to secrete certain chemical attractants. So these attractants, when they are released to the soil, so this rhizobia that is present in the soil, so it get attracted towards the roots of the plant through its nod factors, and it comes and accumulates in the surrounding vicinity of the root hairs of the leguminous plants. So once it deposits near the root hairs of the leguminous plants, so now it results in curling or curving of the root hair. And this rhizobium, once the root hair is curved, so this rhizobia, it forms a small incision or the small opening in the root hair, and it enters inside the root hair, resulting in a formation of the infection thread. And once the infection thread is formed, so this infection thread, it continues to grow along the root hair, reaches the epidermis, and finally it fuses with the plasma membrane of the root cortex. And once it comes in contact with the root cortex, it induces rapid cell division in case of the root cortex, resulting in the formation of the nodules or resulting in the formation of the small tumors. And in case of this root nodules, if we see, so this rhizobia will be present in the form of a bacteroids. So this bacteroids includes, these bacteroids are anaerobic actually. They are sensitive to the oxygen in the environment. So bacteroids will be anaerobic, whereas rhizobia is aerobic organism present in the soil, and they are gram-negative rod-shaped bacteria. So that is regarding the structure of the rhizobia as well as the formation of root nodules. So once the root nodules are formed, so what will be present in the root nodules? So root nodules will be having an enzyme that is called as nitrogenase. So root nodule has the enzyme that is called as, the bacteria will be having the rhizobia that is present there. It will be having an enzyme that is called as nitrogenase, whereas root nodule, so it will be having a oxygen scavenger that is called as leg hemoglobin. So leg hemoglobin will be present in the root nodules, whereas nitrogenase will be present in the bacteria. So what is the significance of this leg hemoglobin that is present in the root nodule? So this leg hemoglobin, it is a pinkish pigment or the pinkish protein that is present, which acts as a oxygen scavenger. So I said that the bacteroids, once they are present in the root nodules, so they require an anaerobic environment or the oxygen-free environment for conversion or for fixing of nitrogen into ammonia. So this leg hemoglobin that is present, it forms the oxygen-free environment because it absorbs all the oxygen and it creates the anaerobic environment there. So that's why this leg hemoglobin, it acts as an oxygen scavenger. And this nitrogenase enzyme, so it is involved in the conversion of nitrogen into ammonia, atmospheric nitrogen into ammonia through the series of reduction process that is taking place in the nitrogenase enzyme complex. And if we see in case of nitrogenase enzyme complex, so this nitrogenase is made up of a molybdenum ferrous protein. So it is consisting of certain cytochrome system which is made up of the molybdenum as well as the ferrous ion. Okay, and molybdenum, it acts as central metal atom in case of nitrogenase enzyme. And this nitrogenase, it catalyzes the conversion of nitrogen into ammonia. So here we can see the reaction. So this nitrogen, it will be fully reduced into the ammonia by utilization of 8 electrons and 8 hydrogen ions. And this hydrogen ions will be donated by the electron donor that is called as NADH. So NADH plus H plus, so it acts as a reducing agent here which acts as a hydrogen as well as electron donor. So in the presence of 8 molecules of NADH and H plus, so this nitrogen, it will be completely reduced into ammonia, and this is an active process, so which will be requiring 16 molecules of ATP for the conversion, complete conversion of nitrogen into ammonia. So it is represented diagrammatically here. So in case of, this is the enzyme nitrogenase, this is the active site. So once the nitrogen, atmospheric nitrogen is, it comes and binds to that of the active site of the enzyme. So the bond that is present, the covalent bond, 3 covalent bonds will be present in between the 2 nitrogen, okay, in order to form a dinitrogen gas. Okay, so this nitrogen, the bond present between the nitrogen, first it will be reduced or it will be cleaved, resulting in the conversion of nitrogen into NH2. Okay, so this, it will convert the nitrogen into NH2 molecules of NH. And this NH will be further reduced to form NH2, and final reduction, it results in the formation of NH3, that is ammonia. And in each process, 2 hydrogen ions are required, so which are donated by the 8 molecules of NADH molecules. Along with that, electrons also will be utilized, which helps in reduction of the nitrogen gas or the atmospheric nitrogen into ammonia. So ammonia, once it is formed, it can either be converted into nitrate or the ammonium ion, so which will be further assimilated by the plants. So what happens once this ammonia is produced in the plant? So in the next part we will be discussing regarding fate of ammonia. So once this ammonia is formed by the plant, okay, it is formed within the root nodules. So this ammonium will be converted into ammonium ion, and this ammonia will be utilized in the formation of amino acids by the 2 different processes called as transamination as well as deamination or reductive amination process. So in case of transamination, the amino group from the one amino acid will be removed and it will be transferred to the keto group of the keto acid. Okay, so keto acid will be containing a ketone as a functional group, and this keto acids are obtained in the Krebs cycle, such as it may be alpha ketoglutaric acid or acetic acid, aspartic acid, as such. Okay, so not aspartic acid, sorry. So pyruvic acid or the alpha ketoglutaric acid. So all these are the different keto acids that are formed during the photosynthesis, during the respiration or the Krebs cycle. Okay, so these keto acids, for this keto acids, the amino group of the amino acid will be added, or the ammonia that is formed will be directly added. So it is a group transfer reaction. Ammonia will be transferred from one particular compound to the another particular compound. So here, if you take the example, so here there are certain examples, like if we consider alanine. Okay, so alanine is one of the amino acid. So this will be reacting with that of the in the presence of the pyruvic acid. So in the presence of pyruvic acid, so this alanine, it gives its amino acid to the pyruvic acid, alanine, or it may be aspartic acid as such. So any amino acid, if it donates its amino group to the pyruvic acid, it results in the one more amino acid formation that is called as alanine. Suppose if amino acid from the amino group of one amino acid is added to the alpha ketoglutaric acid, then it results in the formation of aspartic acids as such. So here amino group of one amino acid will be transferred to the keto acid to form the another amino acid, and this process will be catalyzed by the enzyme that is called as transaminase. So examples for transamination is formation of amides such as aspargine, glutamine, and from the amino acids such as aspartic acid, glutamic acid by the addition of amino group. And fixed nitrogen in case of soya bean, it can be transported as a urides also. So in the form of a urea or uric acid, it can be transported, and later they will be utilized by the plants. They will be translocated and they will be utilized by the required plant parts. So that is regarding transamination. So next is reductive amination. So in case of reductive amination, the glutamate is formed from the ammonium ion by the reaction of alpha ketoglutaric acid. So that is here keto acid, it will be reduced into an amino acid by that of the ammonium ion that is formed in the nitrogen fixation. So as a result of nitrogen fixation, ammonia will be formed. So this ammonia will be converted to ammonium ion, and this ammonium ion will be added to the particular keto acid in the presence of a reducing agent such as NADPH, and it results in the conversion of a keto acid into an amino acid. So that is glutamate. So for example, they have considered alpha ketoglutaric acid as a keto acid. When it is added with the ammonium ion in the presence of NADPH, it results in the formation of glutamate as well as the water, and NADP, that is oxidized, it will be released. And this particular reaction is catalyzed by the enzyme glutamate dehydrogenase. So that is regarding reductive amination. So in reductive amination, directly ammonium ion is added to the keto acid in order to reduce it into amino acid, whereas in transamination, amino group of one amino acid will be added to the keto acid to form the one more amino acid. And this reaction will be catalyzed by the transaminase enzyme. So that is how the ammonia that is formed in case of plants will be utilized. So once the amino acids are formed in case of plants, so this ammonia will be assimilated and it will be utilized. This amino acids will be involved in the biosynthesis of the proteins. So they will be involved in biosynthesis of proteins, and this will be catalyzed through the this will be catalyzed through the process of translation and all, where mRNA will be fixing this amino acids. They will add the amino acid to form a polypeptide chain, and that is how the fixed nitrogen will be assimilated and utilized by the plants. So that is regarding the detailed explanation for the nitrogen metabolism that is seen in case of the plants, how exactly they are going to fix the nitrogen, atmospheric nitrogen, and how it will be utilized by the plant. So it is explained in detail. So that is regarding the synopsis of this particular chapter, mineral nutrition.